Can you all see the screen that I'm sharing?
Yes, sir.
Okay, so no doubts. Yes, no?
No, sir.
Okay, great. No doubts are mostly good signs that you understand everything. So I would like to believe that in good faith. So let's move forward from here. Okay. So I gave you some homework in the last class to quickly go back and look at color blindness and thalassemia, right? Did you go back and look at color blindness and thalassemia from your NCRT? Yes, no? We have
Yes,
Kiran
sir.
joining in. Okay, great. Great. It's good that you people are vocal. Please stay vocal in today's class. Okay, so let's go with Parker first. Parker, would you like to talk about color blindness? Basically, I already told you that it's a sex-linked recessive disorder. So mostly the questions that will come in long answer type questions will be pedigree associated, but some factual 1 marker or 2 markers can also come from these parts. So, and those will mostly be about mechanism. So how does it happen? Some facts associated with that disease, etc., etc. So tell us, what should we know about color blindness? What did you study from NCRT? Parker, if you're speaking, we can't hear you. It's very feeble. You need to increase the volume or something, or come. Can anyone hear Parker?
No, sir.
All right. Yeah, Parker, would you like to join back? Okay, we'll wait for you. I'll go to Aisha till then. Aisha, would you like to talk about, let's talk about thalassemia first then. Would you like to talk about thalassemia? The same things. It's autosomal recessive, but what it is, what happens in this disease? Aisha?
Yes, sir.
Yeah.
So it's an autosomal recessive blood disease, which is transmitted from the parent to the offspring for unaffected carriers.
Mhm.
And this could be caused due to mutation or
Aisha, your voice is not very stable. And Parker, we still can't hear you if you're speaking. Very feeble. I think it's some connection issue. Whatever you're using, if it's Bluetooth, just remove it or do something different that you're currently doing. Because I can hear you, but very, very feeble. Nasha, we can't hear you. Asha, you are mute if you're speaking.
Yes, sir. So
Yeah, great.
this is caused due to mutation or deletion in the gene, which causes a reduction in the globin chains.
Right. Which globin?
Alpha and beta
Yeah,
chain.
the chains of the hemoglobin molecule, right? So in
Yes.
thalassemia, what I'll also keep explaining it as Asha is talking about it. So pay attention here. Okay. So sorry, there's only one L and two S. Yeah. So in thalassemia, what Aisha is telling, so it's a disease of, it's a blood disease, right? And specifically in blood disease, it is associated with hemoglobin. Now you know what is hemoglobin, right? It's a protein, a pigment protein, which also gives red color to the blood. It's present on the surface of RBCs, and it's a very huge molecule. It has 4 subunits. So it's basically a tetrameric protein. Tetra means 4, meric means like there are 4 parts of it assembled together to make it a functional molecule. So it has 2 alpha subunits, right? And it has 2 beta subunits. Okay. Now this alpha subunit and beta subunit you can understand like so let's say this is one protein arm. Protein arm could be like anything long chain. So this is an alpha. On the other side also there will be an alpha chain like this. So 2 alpha. Then you need 2 beta, which I'm going to make slightly different. Let's say this is a beta. And on the other side as well, you will have another beta. So they together join to make one big hemoglobin molecule. And at the center of this molecule is iron, Fe. Okay, that's why iron is important for hemoglobin. You must have heard, right? Iron deficiency leads to hemoglobin deficiency. Because if there is no iron in our body, and this is exactly the same iron which is present in your cars, in your refrigerators, ACs. So iron as an element is the same iron that we need inside our body. Okay, so that doesn't mean we can start eating metal, but there are ways we get all these elements from food. So spinach is a rich source of iron. Okay, so is many kind of flesh are good source of iron. When we eat these things, we get this iron, it's absorbed inside our body in this elemental form, and that's where we use it. Our body uses it to make hemoglobin. Now this iron binds to oxygen. So also another question that comes for hemoglobin molecule, and that you should remember, is that one hemoglobin molecule can bind to 4 oxygen, not O2, but O, 4 oxygen atoms. Okay. It is because this Fe in the core, the iron in the core, can hold on to 4 individual oxygen atoms. And that's how it transfers oxygen from one part of the body, which is lungs, to every other part of the body, which are tissues. Okay. Is that clear, everyone?
Yes, sir.
Yes, sir.
Yeah. Now, because it has total 4 units, 2 alpha subunits and 2 beta subunits, what Aisha was telling is that these units have genes, right? So all these are single, like first produced single through genes, which get transcribed to make these units, and then they are assembled together. So basically in this defect, there might be some mutation or deletion either in the alpha subunit or in the beta subunit. Anyone. And because of which you will have deficiency of either alpha or beta. Okay. And this will result in deficiency of the whole hemoglobin molecule as such. Correct? And that's why it's called, this causes thalassemia. And anything else, Aisha, you would like to tell about thalassemia? Types or something?
No, sir, I actually know.
No? Okay, so in your NCRT only, There are two types of um, thalassemia. It's very easy to understand now. One is called alpha thalassemia. And since you now understand hemoglobin molecule, you all can tell me what is alpha thalassemia? What could be alpha thalassemia? It's the most simplest thing to reduce. Hemoglobin, I told you, has 4 subunits, 2 alpha, 2 beta. So what could be alpha thalassemia?
The alpha chain is different.
Yes. So the gene, perfect. So the gene that encodes for alpha subunit is defected or mutated or deleted. So you don't have alpha chain synthesized, and beta is the gene for beta subunit is defected. Is that clear, everyone? Now, for alpha thalassemia, the genes which are mutated are hemoglobin alpha 1, HbA1, and hemoglobin alpha 2, HbA2. Okay. And both these genes, this is for entrances. You don't have to remember it for boards, but this is present on chromosome number 16. Okay. Will you remember that? Whereas for beta thalassemia, there is just one gene responsible. So alpha thalassemia is more common, as you can see. There are 2 genes, and this is hemoglobin B, simple beta chain, HBB. There is no 1 and 2. There is only 1. And this is present on 11th chromosome. Now, you must be thinking that hemoglobin is one protein. But do you see that the 2 subunits, the gene for 2 subunits that make hemoglobin are present on different, entirely different chromosomes in humans, right? Yeah. So this tells you that it's not like we have numbered these chromosomes from 1, 2, 3, 4, so on. And for the sake of our simplicity, we decided that this one we will call chromosome 1, and it has certain genes. But for any human or any organism to form totally, it is dependent on all its chromosomes, not just 1, 2, or something. So even to make a hemoglobin, you need to have both chromosome number 11 and chromosome number 16 together, functional in a human. And because half part, the beta chains will come from here, the alpha chains will come from here, and then together they will form a big hemoglobin molecule. Okay. Now, you studied thalassemia, you also studied sickle cell anemia. Now, often, how will you, so in sickle cell anemia also, anemia is also a problem, a disease, a blood disease, where hemoglobin is less, or the oxygen carrying capacity is compromised, right? In sickle cell anemia, the difference is, what is the difference there? How will you differentiate between thalassemia and sickle cell anemia? Since now you have studied both of them, that's why.
Thalassemia will be quantitative blood disorder,
Okay,
whereas
that's great. I was not expecting this answer, but yes, that's exactly thalassemia. You said is a quantitative disorder, but what do we mean by quantitative? And what is the other one? What is sickle cell anemia?
maybe it's the quality of the blood.
Qualitative. Okay, so thalassemia you say is quantitative, right? And sickle cell anemia is qualitative. Thalassemia is quantitative and sickle cell anemia is qualitative. Okay, but why is thalassemia then quantitative? What is quantitative versus qualitative? And any example from real life? See, these are all terms, you know. I always say that to understand science, always try to tease it out, tear it inside out, and try to understand what it actually means. Do not just learn because these terms are written in books. Someone could have just made it up. How do you know that what you are studying is true? Just because something is written in NCERT, or just because someone is teaching you that thing, or even just because these things come in examinations and you have to pick an answer, shouldn't be the parameter for you to accept truth. Truth is testable. Truth makes sense. Truth is logical, right? So think about it. What is quantitative in real life? Anything which is qualitative and quantitative in real life?
So quantitative means that the quantity of the blood is reduced.
Quantity of blood is reduced. Let me tell you, neither in thalassemia nor in sickle cell anemia, the overall quantity of blood is less. So that's a myth. People think that the overall blood will be less in the body. It's not like that. Blood is made up of white blood cells, platelets, RBCs, and plasma. So the quantity, which means the volume, is often decided by the plasma content and everything dissolved in it, along with and the water. So blood is not something very special, a liquid. Like most of the liquids, like milk, blood is also around 94% water only. Okay. So the quantity, if it is reduced, that's another problem of not making enough blood in someone's body. Then it's the problem of the bone marrow, which produces blood. But here, that's why I asked this question. So sometimes we understand something wrong, but we just know the right fact. Okay. So that's important to understand it correctly. So forget about blood. Okay, Hiro, for a second. And tell me one example from your real life where we can understand quantity and quality, a qualitative assessment, a qualitative measurement, and a quantitative measurement properly. Any example? Anyone? No example? It's a mathematical reference, mathematical thing. Quantity and quality. So suppose you're taking this class, okay? And suddenly my voice starts breaking up. So, and I tell you that, oh, I think the quality of my internet connection just deteriorated, right? Can I say that quantity of my internet connection just deteriorated?
No.
No, why? Why I can't say that? I can actually say that if I open and test the internet latency or the speed, the upload and the download speed through there are Wi-Fi speed checkers. So they will tell me exact number that my download speed is, let's say 20 Mbps. So if I have that data, I will say that earlier it was 60 Mbps, now it is 20 Mbps. So it's quantitatively it has decreased. That's why the quality has also decreased. Does it make sense to you?
Yes, sir.
A quantitative problem is defined. You can define it in grades, like it's 10% decrease, 20% decrease, increase. So either there is a decrease or there is an increase. Qualitative, on the other hand, is about the quality, even if the product is same, okay, but the quality is different. For example, I give Rabia 2 apples and I give Iram 2 apples. Both of them tasted those 2 apples. And Rabia said that the quality of my apples are better than Iram's. Now, quantitatively, both have 2 apples, but qualitatively, maybe Rabia's apples could be freshly plucked from the farms, and Iram's apple could be, very, very old and have some problem, or is not that sweet, or is a little sour. You understand what I'm saying?
Yes,
Yes,
sir.
sir.
Yeah. So in sickle cell anemia, it is not the quantity of RBCs or hemoglobin that is compromised. It still has the similar number of hemoglobin molecules, but all these hemoglobin molecules are either incorrectly formed, so they are incorrectly functioning molecules, or these RBCs are incorrectly shaped. So if you will count, you will see that, okay, in a sickle cell anemia patient also, the number of hemoglobin, the number of RBCs look the same, but all of them are sickle shaped. So they are incorrectly formed RBCs. Whereas in thalassemia, you will see that the number of hemoglobin molecules themselves decrease because you do not have a functional beta or alpha chain to make up the molecule. That's why thalassemia is a quantitative problem and sickle cell anemia is a qualitative problem. Does that make sense to all of you?
Yes, sir.
Anyone who wants me to explain this again can just write in the chat. Because yes, okay, great. Parker raised hand. So Parker, you want me to explain this again? So okay. So think of this problem like this. So in thalassemia, we need basically 4 components to make a hemoglobin: 2 alpha chains and 2 beta chains. Total 4. 4 components are required to make one functional hemoglobin molecule. In thalassemia, these components are not present. Either alpha is not present or beta is not present. So it's like you want to ride a bicycle or you want to assemble a bicycle, but all the parts of bicycle are not present. That's why you cannot assemble the bicycle. That's the problem of quantity of parts. So since alpha chain will not be present, you cannot put 4 beta chains and make a hemoglobin, right? It won't happen. So you need 2 alpha and 2 beta to make one hemoglobin. So similarly, you need 100 alpha and 100 betas to make how many? 50 hemoglobin molecules, right? But if because of mutation or deletion, you do not have these parts in enough numbers, then the quantity of hemoglobins produced in the body will be less. So thalassemia is a quantitative problem because the number of hemoglobin molecules decreases. Okay. So humans cannot, person suffering cannot make enough hemoglobin molecule. On the other hand, in sickle cell anemia, everything will be available. You will assemble a bicycle, but these parts are not functioning properly. They are available. So your tires are flat tires. They don't have air inside. Your chain is jammed. It's not functioning properly. You know, the spokes of the wheels are somewhere broken or bent. So the wheels are not functioning properly. The handles are bent, but every part is available. So it's not a quantitative problem. It is a qualitative problem for those parts. The quality of the parts are not good. So in sickle cell anemia, the number of RBCs and the number of hemoglobins might be the same, but still not functional. So it's a qualitative problem. Does that make sense, Parker? Do you understand? Yep. Everyone understands? I want thumbs up from everyone if you understand this. Perfect. So write down thalassemia is a quantitative blood disorder, whereas sickle cell anemia is a qualitative blood disorder. Okay. Okay, perfect. Now let's finally go to the last one, which is color blindness. Yes, Iram, would you like to talk about color blindness?
Yes, sir.
Okay, before we go there, it just occurred to me to ask. Sorry, I forgot it. I didn't ask it because I normally use a lot of colors on the board. So any one of you have any difficulty differentiating between any colors? You can just write to me in the chat directly. I should be knowing that if any one of you has problem differentiating between shades of red and green or red and yellow or blue or something. All right. Okay, perfect. Cool. Yes, Iram, over to you. What is color blindness?
Yeah. So firstly, color blindness is sex-linked recessive disorder.
Mhm.
It is inability to differentiate between certain colors. That's
Mhm.
what color blindness is.
That's what called color blindness. You will also hear a term or you must have read a term called night blindness, right? Night blindness is people where people cannot see in the dark at all. Like whenever sun goes down in the night, they cannot see. It does not mean, so again, there are myths associated with this night blindness. They think that after sunset, they cannot see at all. It's not true. If they're in a room which is lit, like I am sitting in a room which is lit, they will be seeing everything okay. It's just like below one threshold of light, they won't be able to see. And can you now think and figure out why? In your juniors classes, you must have studied about what helps us to see color, etc. Right? In the eyes.
Retina.
Perfect. Retina is the kind of a membrane where all the light falls. But what is present on retina that helps us see?
So you're asking about the cell?
Yes, I'm asking about these are neurons or photoreceptor cells. Remember these term? Even if you don't remember this term, let me just simplify it for you. So photo means light. The packet, the unit of light energy is called photon. Okay. So one photon is one packet of light energy. It's very small. Photo is associated with light. So when we click photo, we are actually capturing light. That's why it's directly called photo. Photoelectric effect is where light produces electricity. Photosynthesis is where in the presence of light, plants synthesize their own food. Photoreceptor cell is simply the cell in the eyes of human which receive light, photoreceptor, and then sends the signal to brain and we can then see. Okay, so these are basically neurons and there are two kinds of photoreceptor cells. One are called rods. Remember these? One are called
Yes.
cones. Yes. So do you remember what does what? Which one is responsible for what? Anyone remembers? What do rods do? What do cones do? And they're just called rods and cones because of their shape. So rods are shaped like this. Cones are shaped like this. So they have a cone, okay? Whereas this is like a rod. But what do they do? Any idea, people?
Rods are responsible for vision at low light.
Okay, rods are responsible for vision at low light, correct. So I'll tell you one quick way to remember this. C for color and C for cones. So cones provide you color vision. Okay. Will you remember that? Yes, no, something? Yeah. So cones provide you color vision. And for color vision to occur, we need light because light should fall on something and come come to our eyes for us to be able to see colors. So that color vision is dependent on daytime or lots of light intensity. So cones give us color vision, which is also called as daytime vision or daylight vision. Okay. So it's light based vision. So we need light. So cones are mostly functioning whenever you are in a lit environment. Okay. Rods, on the other hand, like Iram rightly pointed out, it gives us low light vision. So in low light, it must have happened with you as well. When we sleep, even if there is a very small light source in the room, which there should be. Actually, we do not sleep in a pitch dark room. Any one of you who sleeps in a pitch dark room, like you open your eyes and you can't see anything? Anyone
Yes,
who
sir.
does that? Yeah. So yeah, you should not do that actually. There should be at least, okay, I don't know. Okay, all of you do. Okay, cool. But try not to, I'm not also saying that put a lamp on or something, but so what I do is while sleeping, I keep a small light source like a fairy light or something on in your room, which is very, very lower than your threat. And it should not be a blue light. It should not be white or blue light, the source. It should be yellow or yellowish light, basically yellow red, something. So a very small amount of light should be coming wherever you are sleeping. And that's because when you wake up, your rods are actually functioning because you can't see. But has it happened to any one of you that you woke up and you can still differentiate that, okay, that image is like the almirah and this is where the table is and that moving thing is the curtain. So you can differentiate between objects because you see different shades of gray and black, but you cannot see color of anything, right? Has it happened to you in the night when you wake up in your room with no light?
No.
Yes or no?
No.
No, because yes, Iram, it has not happened to you because you sleep in a pitch dark room. So you need you need at least some very, very minuscule, but some source of light for the even for the rods to function. Okay. So it has happened with Parker. So Parker, I believe, sleeps in slightly um, dim lit room, which we all should do. Um, our ancestors used to sleep even whenever they used to sleep, even if it was in the cave, there should be something, a burning coal or something around, or fires in the cave, a very small fire at the entrance. Or if you are sleeping under the sky, all animals that sleep under the sky always get some light signals from the stars and the moon's light. So basically, only the organisms that sleep in the burrow, like rodents, like mouse, snakes, reptiles, and amphibians, um, they sleep in pitch dark. Mammals often do not sleep in pitch dark. Humans are mammals, so we should not sleep in pitch dark room. So rods are activated when there is very dim or low light. And in this case, we can only differentiate between objects, but we cannot see any color, no color perception. So you can write here for rods, no color vision. So it's basically black and white vision. Okay. And this is color vision, cones, color, rods. How I remember rods for night vision is rod is like a torch, you know. So a torch is a rod. So you need a torch in the night. So you need rods in the night. And you can see colors in the day. So you need cones in the day. Okay. Perfect. Now, in color blindness, now you must have, yes, Iram, would you like to take forward from here? I made the job a little easier for you.
Yes, sir. So, color blindness is caused when two of these cells fail to perform their function.
Two of these. There are two only. The two of what?
I
Two
mean,
of which cells?
rods and cone.
So rod has nothing to do with color. Why do you think if rods fail to perform, you have night blindness? That's what I was trying to tell you. That's what the whole story was for. So if someone's rods are all defected, so there is a mutation in all the rod cells, but the cones are working perfectly, that person will see perfectly in the day when there is lots of light. But the moment the light goes beyond a limit, and when it is difficult to differentiate between colors, the person will basically go blind. Okay, like someone who's sleeping in a pitch dark room, so you will not be able to see anything. So rods are associated with night blindness. Cones are associated with color blindness. So what Iram is saying basically means that there are 3 types of cones. Okay, and these 3 are the elemental. They are called RGB. So cones can only uh, um, perceive red, green, and blue spectrum. And that's enough to see the world and all the colors in it. And it will make sense to you. Mariam say, Parker has a question. What if, so first of all, you people don't have to address me by saying sir, okay? Especially when you're writing, you can just not write sir. I'm trying to get rid of it even when you're speaking, but I understand that's difficult because you are conditioned to call anyone who teaches you sir. It will take some time to get out of your vocabulary, but don't write it. Okay. Yes. So what if both are not present? Then the person is blind. If both rods and cones are not present, which means the retina is not functional and no photoreceptor cell is working, then the person is a blind person. So that's what most of the person whose brain is okay, but eyes are faulty, like this is what happens to them. Their rods and cones are not functional. So the retina is not functional. So light comes in the eye, falls on the retina, but no receptor cell absorbs it and sends a signal to the brain. So they can't see anything. Okay. Does that answer your question, Parker? Total blindness. Yeah. Now, cones are of 3 types: red perceiving, green perceiving, and blue perceiving. And these 3 are called the fundamental colors, RGB. Your mobile screens, your laptop screens, all the screens basically have these 3 kind of light emitting diodes only. And using just R, G, and B, you can make any color which is present in the whole spectrum. Those of you who have a skill for painting or who like drawing or playing with colors around know that these 3 are enough to make any color, right? In different proportions. So RGB, red, green, and blue, are called elemental fundamental colors. Let me tell you that the cone which is for blue is most stable. So mostly people are colorblind for red and green. This is the most common color blindness, where they cannot differentiate between red and green or the shades of red and green. But most of the colorblind people can see blue very well. How many of you know that there is a reason that Facebook is blue? I hope all of you use Facebook, right? You use Facebook, Rabia, Aisha, Iram?
Yes, sir.
Yeah.
No.
No, okay, cool. But have you seen the logo of Facebook or the page of Facebook? It's a social media platform. It's blue because there are reports and people believe that Mark Zuckerberg himself has some issues differentiating between red and green very prominently, but blue is what most of the colorblind people can see. So even in presentations where you are keeping in mind that colorblind people can still see everything, one is advised to not use shades of red and green together. Okay, but you can use different shades of blue, dark, indigo, and different to show your data, things like that. Okay. Yes, so Aisha knows it. So Zuckerberg has problems seeing red and shades of red and green. So he made the whole Facebook blue. That's a very, it's a very, I don't know if it's thoughtful, but it's convenient for him to do that.
So
But yes,
So isn't yellow a fundamental color rather than green? Because you can obtain it from
No, you can
yellow.
make yellow by using red and green. You cannot make green using red and yellow. So fundamental color is which can give rise to all other colors. RGB is the fundamental color. If you mix the right proportion of red with the right proportion of green, you will end up making yellow. But you take yellow and red, you won't be able to make green.
So you take yellow and blue.
Yellow and blue, and you can make, yeah, right. But not all shades of green. But RGB is fundamental color in biology, like in perception of organisms. Okay.
Okay.
With these three colors, you cannot make white or black. Let me tell you. But because white and black does not come in the spectrum, white means presence of all colors, black means absence of all colors. So even someone can argue that, along with RGB, you need black and white colors also if you want to paint. But that's a different thing. You understood what I'm trying to say, right?
Yes, sir.
Okay, good. So red and green color blindness is the most common, but it's not like blue color cannot have mutations. In biology, everything can be faulty. So there are reports and there are recorded cases where people have difficulty differentiating blue, but those kind of genes will not prevail and will not be selected in nature, because the problem is every organism has to perceive blue, because water reflects blue, the whole sky is blue, and the most what do you say, diffracted color in our atmosphere is blue. That's why we see space as blue. Space is not blue. It only looks blue from Earth, the sky. If you go to other planets or you go in space, it is pitch black with stars studied in long distance. So it's not at all blue. It just looks blue to us because the sun rays, when they come, the blue gets diffracted the most, and it's everywhere. With blue, another important and interesting fact is you will see a lot of a variety of insects, birds, and animals which are blue. If I ask you a bird which is blue or has blue feathers, you will very quickly tell me which bird is blue or has blue shades, blue feathers. A peacock, right? Peacock has bluish feathers. Anyone has seen peacock?
Yes, sir.
Yeah. Okay.
Yes, sir.
And many insects are also blue. They look blue, right? But you will be surprised to know that none of them can make a true blue pigment. Blue pigment is very, very, very rare in biology. So how all these creatures look blue? They look blue because of a physical phenomena called where light reflects at different angles. What it is called? Diffraction. Diffraction. Yeah. Diffraction and iridescence give rise to blue color. So these surfaces are not blue, but when light falls on it and gets reflected back to you, most of the wavelengths gets absorbed, only blue comes out. So you think that they are blue, but they are not blue in reality. Okay. And how you can prove it? Again, never believe anything which is said by anyone. You take a peacock feather that looks blue. There's a question by Aisha in the chat. No question is weird, first of all, Aisha. And is it possible that maybe if I see a thing and that's yellow, but someone might see the same thing and say it's yellow when it's actually red for them? Yes. So what you're trying to say is different color perceptions. It's not, let me tell you, it also happens in normal humans. So if I show you the same color, maybe in the next class, I'll show you some optical illusions, but I'll show you the same color, but in different backgrounds, and you will every time think that it's a different color. Okay. Because our perception of color depends on the contrast of in which we are seeing that color. There's a very famous internet post. If you people would like to go and search for it, it's about a dress, a women dress that's actually colored golden and white. But when it was asked by people, so someone was wearing it, and a photo was clicked, and she uploaded the photo, and people were asked, "What do you think is the color of the dress?" And most of them said black and blue, whereas it was white and golden. But the gold part, they started seeing as black, and the white part they started seeing as blue because of the contrast in the background in which she was standing wearing that same dress. So the same dress wearing in two different contrast you will think that these are two different colors. So color perception is a very, very interesting thing, which lots of scientists are working around in the world. Yes, so the lighting also matters, right? Yes, you are perfectly correct, Mariam. Parker, the lighting matters a lot. So Aisha, the answer to your question is yes, it's very well possible. So in color blindness, the red or the green cones, so 2 out of 3 that Iram was talking about is these two, red and green out of these 3 cones are defective, and because of which you cannot differentiate between red and green color. So NCERT only talks about red and green color blindness, but just remember that blue color blindness is rare, but it is there. Okay. Okay, perfect. Anything else you would like to tell us about this, Iram? And then we'll move ahead.
No, sir, nothing else.
No. Okay, so I'll tell you another thing. Remember, just like any sex-linked disorder, this disorder also is on the X chromosome. So it's X-linked disorder. And for a female to be colorblind, she needs both copies defected. Both the X chromosomes should have defected copy or mutated copy for these genes. And for a female to be colorblind, the father of the female must be colorblind, and the mother should at least be a carrier. Yes or no?
So,
So only if the father is colorblind and has this genotype and it's crossed with the mother who's at least a carrier, then only a female child can be colorblind, right? So it's rare to get a female colorblind. Okay, that's why I asked in the beginning, since all of you in this class are females, if any one of you would have been colorblind, then biologically, your father must also be a colorblind person, and your mother should be a carrier. That's a very, very rare event to happen, but it happens. And why does it happen? Because color blindness is not a lethal gene. Even if a father is colorblind for green and red, and the mother is a carrier, mother will not know. Mother will be completely normal. She will be able to differentiate between red and green because a normal X is there, but she will carry a mutated copy. Father will not be able to differentiate between green and red. But since it is not a lethal problem, you can survive even if you're colorblind, right? So that's why there are females in the world who are colorblind, but they are only 0.4% of the world's female population. So there are 0.4% of females in the world who are colorblind. In terms of numbers, it's a huge number, again in lakhs, but it's there. But for a male to be colorblind, it's very easy. The mother either has to be a carrier or a colorblind person. So even if a mother is carrier, half of her sons could be colorblind. And again, it's not lethal. So this kind of people can stay and do very well and live and no problem. So there are around 8% of males in the world. 8% is a huge number. Let's assume that the world has equal males and females, 4 billion males and 4 billion females. So 8% of 4 billion is a huge number, right? So they are colorblind. So again, if they ask you a question, why it is rare to find a colorblind female, you can give the same logic that you gave for hemophilia. But in this case, it's possible for a colorblind female or a colorblind male to go on and give rise to progenies and pass it on, because it's not a lethal disease. It's not a lethal mutation. Okay. You just can't do certain jobs. You cannot apply for certain jobs if you're colorblind. Okay, perfect. So these were all genetic Mendelian disorders that you had to cover in this chapter. And apart from that, I already told you about chromosomal disorders. So in chromosomal disorders, in the last class, I told you whether either there is aneuploidy, which is gain or loss of a chromosome, or there is polyploidy. Polyploidy is not tolerated by humans or animals. So there is no polyploidy disease. It's not possible. It's tolerated in plants, and sometimes it causes some problem, but most of the time it does not causes any problem for the plant growth and development. In fact, we use this property of plants to generate genetically modified and more nutritious and enriched plants. So for example, rice. Your rice can have, there are more than 50,000 varieties of rice in India alone. I'm not talking about the world, just India. All of them are not grown because all of them are not, some have a more water tolerance, some have more drought tolerance, some have more nutrition and all. So few varieties are grown all across, but we have more than those varieties. And some rice varieties can even have 6 copies of their genome. So they are hexaploid. Tetraploid rice is very common. Hexaploid rice is also there, where vitamin enriched. How many of you have seen the rice packets that come to your home? Have you seen the rice packet? Sometimes they mention that this is a vitamin and iron enriched rice. Yes, Maria. Yes, Parker. Did you see anything like this on the rice packets that come to your house? Vitamin A enriched, or protein enriched, or iron enriched rice varieties, because they exist. Yeah. So rice basically is a good source of starch. It does not contain a lot of protein and vitamins, but nowadays scientists have created these varieties, which helps the society to get these nutrients effectively. Okay, so only thing which is possible in humans is aneuploidy, which means gain or loss of a chromosome. And in chromosomal disorders, you have to just remember 4 disorders. Okay. First is your Down syndrome. Okay. Now, Down syndrome is just a trisomy, like there is one extra copy. So there should be a disomy. Di means 2 copies of every chromosome should be present in our body. One from father, one from mother. Trisomy is 1 extra copy. So Down syndrome is trisomy of chromosome number 21. Okay. So it's called trisomy of 21, and it causes Down syndrome. And because of this, it was actually found by, or discovered, not discovered, but characterized by Langdon Down. It feels a bit weird to me that people who found diseases, the disease got named after them. It's like Down is a surname, but now it's a disease, Down syndrome, because Down figured it out. You'll also find Turner syndrome, Klinefelter syndrome, Alzheimer's. Many disorders are named after their characterized, like doctors and scientists. So Langdon Down figured it out. I don't remember the year, but it was somewhere in around the mid 19th century. So 1850s or 1860s. Figure it out from your NCERT. It should be some 18XX. 60s more. Some 6 is my guess. Anyways, so Langdon Down did it. And more important are the symptoms. So the symptoms for Down syndrome is mental retardation. So babies are not developed properly mentally. Their brain is not developed, and they have a short stature. Their body does not develop as well, and the body is it remains short as they grow in age. And they have a smaller head as compared to other babies. They have a smaller head when they're born. So that's called a small head syndrome. And as compared to their body, the body itself is short. And sometimes in the most severe cases, so mental retardation, a small head, short stature, and sometimes in very severe conditions, they have a furrowed tongue. So the tongue, if you see your tongue carefully, you know, when a human is formed, our face, when it is formed in the womb, basically the left part of the face and the right part of the face does not form like this. It forms like this. So there comes a time when openings have to be stitched. So our forehead is already stitched, the nose is formed, and all the lower part, including the lips and the chin, has to be stitched together. There is also a disorder where it's called a furrowed lip. This cleft between the nose and the lip, you will see it's like a groove. Everyone has this groove. This basically is it's stitched afterwards during embryonic development. This remains unstitched, so their lips are like furrowed. Then later on after birth, you can surgically stitch it back, doing surgery. But similar thing happened with the tongue. Our tongue is also, it stays furrowed. So it's like a snake's tongue. What is stature? Parker, stature is height. So as they grow, a kid's height should also grow up. But the kid with a Down syndrome stays small, looks like a dwarf kid. Okay. So that's stature, short height. Okay. Is that clear? Yeah. And so these are the most common and important features you should remember, characteristics or symptoms. The second is Tau syndrome. I'm not sure if this is in NCERT or not, but it's called Tau syndrome. This is again a trisomy, but this is asked in a NEET exam sometimes. So I thought I'll just tell you, this is trisomy of chromosome 13. Okay, similar like Down syndrome. And even the symptoms are very same. Mental retardation is one, problems in speech, and furrowed tongue is not there, but short stature, problem in speech, mental retardation is there. Okay. And then the third one, which is important and is related with the sex chromosomes, is Klinefelter syndrome. So Klinefelter syndrome is basically also known as XXY syndrome. So there are two copies of X in a male. So XY should be the normal genotype. Okay, Rabia has a question. But then there are some people with Down syndrome that are of normal height, right? Yes. So as I told you, all these symptoms may not be present together in the same person. So Down syndrome also has a degree of severity. Most of the people who have Down syndrome does not have a furrowed tongue also. But if the Down syndrome, the severity is too much of the syndrome, then you will see most of these symptoms together. So when we talk about symptoms are sum total of all the symptoms that are shown in different individuals that are suffering from the same disorder. Some might have short stature, some might have a normal stature. Very rarely you will see people with Down syndrome who have a very tall stature. Okay. I personally have not come across any case or any documented case where someone is like, tall. What we consider as tall is a 6 foot height and still has a Down syndrome. But they can have an average stature also. It doesn't need to be short statured all the time. Also, doesn't have to be furrowed tongue all the time. And also the severity of mental retardation is also different. Some kids suffering from Down syndrome can speak certain fragmented sentences, can express what they want, what they need, can also reply back, but some cannot. Does that answer your question, Rabia? That's a good question, actually.
Yes, sir. Thank you.
Yeah. And remember that always holds true for all kind of syndromes and diseases in humans. Take the example of COVID. Did COVID hit everyone the same? No, right? Some people were severely ill with the virus. Some people were asymptomatic. They did not even show symptoms when the virus was inside their body. What it could mean in a long term, we are still figuring out. So now researchers have figured out that people who showed and who did not show severe symptoms during COVID, the severity of long-term COVID effects will be different because the virus, even if it's cleaned by the system, it's not completely cleaned, and some remnants of the toxins stay in the body. But that's a different thing. So for any disorder or disease, it's never uniform, the symptoms. Okay. Cool. Thanks for bringing up the question. So Klinefelter is where there is one additional copy of X chromosome. Now, because of this one additional copy of X chromosome, remember the karyotype now will be changed. You know what is karyotype? Karyon words comes from nucleus. Karyon means nucleus, and type means like, genotype, phenotype. Similarly, karyotype, basically the morphology of the nucleus, not morphology, but the status of the nucleus, where you can see these chromosomes. The karyotype now becomes 47. So you will see 47 chromosomes in this person, 46 normal plus one extra X. And it's also written as 44 autosomes plus XXY. So 47. Okay. Now, because of this extra X, this male body will start showing a feminine development. Okay. So in this, what happens? Write down. These individuals with Klinefelter syndrome, these individuals has overall a male body because Y chromosome is there. Remember, I told you if there is a Y chromosome, the body will develop into a male body, which means it will have male genitalia, male sex organs. Even the musculature will be there. So they will be masculine and all, but they will start showing female characteristics as well. So they will be masculine, tall, but with development of mammary glands for that matter, like breasts will be developed in these individual, and that's called gynecomastia. So one of the classic symptom of Klinefelter is a male who has gynecomastia. The word gyne is always associated with female reproductive system, gynecologist, right? So gynecomastia is development of breast.
So,
Yes.
so is this syndrome present in transgenders?
No, actually, transgenders are they sometimes can be different. It's not true that all yes, many people in the society who are suffering from Klinefelter syndromes end up, being the transgender, like belonging to the transgenders community. But transgender is something. So we all are cisgenders. Cisgender simply means, so cis and trans, it's a word you must have studied in chemistry, right? A cis arrangement of a molecule and a trans arrangement of a molecule, right? Have you studied that while studying functional groups?
Yes.
So if
Yes,
on a
sir.
carbon, there is one O here and there is one O here, then it's a trans configuration, sorry, cis configuration of O because they're on the same side, right? Trans is the opposite. So transgenders basically a body develops. So the half of their characters are feminine and half of their characters are masculine. So these will be female bodies. So they will have ovaries, vaginal canal, uterus, but they will start showing male symptoms also, which is because of which is beards and mustache and a very high coarse voice, or the other way around, a male genitalia organs and will start showing feminine characters, which is Klinefelter. So it's a part of it, but not the whole of it. Does that answer your question, Iram?
Yes, sir.
Yeah, so Parker is asking, can this happen with female? Yes, if this happens with females, then there has to be, it's rare, and you will understand why. See, a female is already XX, right? Now for female to show this kind of a syndrome, she needs a Y. Now, Y is something which was not present in a female, but then where did it come from is the question. Okay, so in other case where it is X and Y, an extra X can come because of error when the cell is very early, when one zygote is becoming two, this X chromosome gets copied in a defected manner. And in the next cell, it could be XXY or even before. So two cells get XXY. Now from this point onwards, it started as a normal zygote, but because of copying errors, it became a Klinefelter syndrome patient. So the Y was already there, X got copied. So this is a more probable scenario. Does that answer your question, Parker? Yeah. But as I told you, in females also, there are other ways by which can happen, which we consider as transgender community. Okay. So, it's actually a very, very important topic that you brought up and a convoluted topic that everyone needs to understand, but the understanding is very limited, especially in society because they don't understand the biology behind it. So it's not just about genders. So gender is one thing. Gender is decided by your sexual organs, right? So you can be cis, we all are cisgenders. So our genders and our personalities just match exactly. If it's a transgender, the gender and the personality will not match. So it's trans. There could be different sexualities as well. That's the concept that people are trying to fight very hard for to get equal rights for. That's called sexuality. That's a very different concept from biological Klinefelter and Turner syndrome. That's where the body is normal. The body has one gender, it's a male or female, but the personality or the hormonal regulation from the brain to the body is different. So it's like a male stuck in a female body or a female stuck in a male body.
So
Now
is it
yes.
intersex?
It's not intersex because intersex also is intersex is I think intersex is more like transgenders. But I'm talking about homosexuality. So we are heterosexuals where the kind of sexual organs, so suppose if a person is heterosexual, so the kind of sexual organs that one person has, the attraction for that individual will be for a different kind of a sexual organ. So that's called a male attracted to a female, which we consider as the norm of the society, is a heterosexual society. But a homosexuality is, again, you have studied hormonal regulation of the body in class 11th, right? That's just the basic that you studied. Remember, hormones that govern development of like mood swings or masculinity, or it also gives you personality. So testosterone is the reason that males in the society behave like males. So with testosterone, there is also, there is one condition called XYY. How many of you know about it? An extra Y. And this kind of syndrome is called the criminal syndrome because too much of testosterone, too much of arrogance. So testosterone always gives any personality a touch of arrogance and violence. And it makes sense in biology because males always used to fight for territories. All males, like, you will very randomly see females fighting for territories. Females defend territories, like lion and tiger females. They defend territories because they have to defend their cubs. That's a motherly instinct. But males have an instinct of fighting for territories, and human males are no different. All the war and the borders and the countries, everything that has been happened. Tell me one one army general from the past who was a female and who started a war on other country, or who wanted to be victorious. You will always find these names, all males, from Alexander to Hitler to all the conquerors were males, right? There's a reason for it, because testosterone drives that kind of a behavior. If it is too much, it drives it unnecessary, and these males they often become offenders. They do violent criminal activities. So hormones can decide our personality, that we know. It's a truth and fact in science. Similarly, if you have one set of sex organs, but a different set of hormones governing with those sex organs, you will always feel indifferent. So society will tell that, okay, you're a boy because you have a penis and you have scrotum, you have testes. But the problem is the amount of hormonal regulation that should make you feel like a boy is not there. So you psychologically, and because of your hormones, feel like a feminine more. So you identify yourself as a female, but you're stuck in a male body. It's very difficult to understand for someone who does not understand the details. And even scientists and researchers do not understand it fully. A lot of things are under research. So people used to believe, since we are talking about Klinefelter and Turner, okay, so there's one question. So the people that have extra testosterone don't have control over their aggressiveness. Most often, physiologically, no is the answer, but that's not very simple. Testosterone levels can also go up when you exercise. So you always have to channelize testosterone. So if a male has more testosterone than required, then the male should either be a fighter, the male should either be a wrestler, the male should either be an athlete who can channelize that energy into some other thing. If the male is a normal person living in society with an extra testosterone surge because of some issue in the genes, then the male will always be more aggressive. It's not just for males, even females. So female athletes who train themselves harder oftentimes have matching levels of testosterone as of a normal male in the society, but their testosterone does not make them males. Their testosterone give them musculature, strong body, like weightlifters, boxers who are females, or athletes, high jumpers, runners who are females. They will have a higher testosterone level in their body than normal females. Does that make sense, Rabia?
Yes, sir.
Yeah. And yes, but the thing is, whenever we have to study something which is fundamental in biology, we have to go to other organisms as well. Humans are not very special. We just have this ego, human-centric ego, that we feel that we are very special on the planet. We are special in a way that we can do a lot of things that other animals cannot. But biologically, we are not very, very special. There are organisms who can do far better jobs than us, but not together. For example, we are like jack of many traits, but master of none. We are not the fastest of the organism. We are not the strongest of the organism. We cannot echolocate with our brain like bats can do. We cannot sense sonar. We cannot sense a lot of other things. We cannot communicate in infrared or ultrasonic waves. We cannot see ultraviolet, infrared spectrums. We can only see very limited. So we can do a lot of things, but everything average. That makes us special. We can climb, but we are not the fastest climbers. We can lift weight, but we are not the best in lifting weights. Gorillas and chimpanzees can do it better than us. We are swimmers, but we cannot swim better than many mammals. So you understood what I'm trying to say. But this behavior of homosexuality, heterosexuality, even intersex and transgenders is also present in other mammals, even amphibians and reptiles. So it's a very innate thing in biology. But of course, because it does not have an ecological benefit, a reproductive benefit, it will never become the norm of the whole population. So it can never happen that 60% or 70% of the population can show a homosexual behavior, because that then will seize down the reproductive rate of that population and it will decline. Same happens for frogs and other species that show this kind of a homosexual behavior that humans do. But they're all either hormones or at a physiological level, it is changes in the genes. Okay. Does that make sense, everyone? So we should be aware, we should keep understanding these things in detail, not just discriminate because we don't understand. There was a time when people used to discriminate between males and females. And since all of you are females here, let me tell you a very interesting study. A white European scientist once, he was a very decent researcher, but once what he did, just to prove that white skinned are superior, he did a quantitative analysis where he took the skulls of white men, like who were dead, from their graves and figured out their volume and compared it with other races, their volume in the skull. And he said that he compared a white man's skull with a black African female's skull and just said that, white are superior because they have bigger brain capacity. So their brains are bigger. But that's not the true representation. He did not compare a white man with a black man. He did not compare a black woman with a white woman. So that's where, and even in males and females, people say that there are certain differences, and those who do not understand the biological basis of those differences end up discriminating females against male and believe that males are more intellectual and males can do more of like scientific work, whereas females are more emotional and they have better motherly instincts. But that's not actually biologically true. We now know that there are amazing fathers in biological systems. For example, seahorse, hippocampus seahorse, and many other species where fathers take care of the eggs or the babies, whereas mothers don't. And we also know of species now where mothers are the more ferocious ones, whereas the males just live on what the hunters are the females, and males are just there for reproduction. So if we do not understand biology and how it works and what are the differences, we will end up being discriminatory. So same happens for topics like transgenders, the third gender, different sexualities, inclinations, because we don't understand them actually. But that shouldn't be the case. That's what I try to tell everyone. So that's Klinefelter's XXY. In a male overall masculine body, you see feminine development. There is something called Turner's syndrome as well. So Turner syndrome is opposite. Instead of karyotype 47, here you will see a karyotype which is 45. So this is where only one X chromosome is present. The other is absent, neither X nor Y. So in books, we often symbolize it as X naught. It's not a different sex chromosome. It's no chromosome. It's like X naught. So because of X naught, you will see now that the karyotype will become 45. And it's written as 44 autosomes, which are normal, plus X. Okay. So what do you expect? Okay, I forgot to tell you one thing. In Klinefelter syndrome, even if the body is male, you will see that the sexual organs also develop as males with secondary sexual characters of a female, but the person is sterile. They cannot reproduce. So this extra X somehow does not allow this XY pair to go on and do everything normal because hormones change a lot. You know that for development of mammary glands and breasts, we need estrogen, right? That's why female bodies also start developing mammary glands post puberty. Make sense now? Are you connecting what we studied in chapter 4? Sorry, chapter 3. So if a person has one extra X with a Y, it will always hinder the development that Y can give to the body. And that's why they are sterile individuals. Okay. And if someone is suffering from this, we just, kind of cast these people out from a norm of the society and do not treat them with respect. So they end up becoming the part of the third gender, which is the transgenders. And they cannot have their own offsprings. And most often they also do not have rights, basic human rights in many countries, which is very sad. Anyway, so in Turner syndrome, the karyotype is 45, and this happens mostly in females. So the overall body, it's feminine body. And you can understand why it cannot be a male body overall. Tell me, now you understand biology. Why it can't be an overall male body with some defects? Why it has to be a female body?
There's no why.
Exactly. Y is very important to give any maleness. If there is no Y, no maleness, zero maleness. So and remember very early when I started human reproduction, I told you that the default state of an embryo is
Female.
female body, right? Even if an embryo has X and Y, if I somehow silence Y or just some genes, and it has been done in some insects, which also have XY sex determination like humans, the research has been done. If you silence some genes and if they do not signal at the right time, the body with X and Y develops into a female body. So X naught will also be a female body, but these female, they will not have male characters, no male characters, zero male characters because no Y, but they will be underdeveloped females. Okay, so their ovaries won't be developed, rudimentary ovaries, and the birth canal, the uterus will not develop properly. Their menstrual cycle won't be proper. Their mammary glands won't develop the way they should. So they will always be a short-statured, underdeveloped females. Okay, write down. Rudimentary ovaries. Yes,
So if you like silence the Y chromosome, then it turns out to be a female. But like, is that allowed to do? Research.
In insects, yes, in insects it is. In humans, we cannot do any research on humans. There are human ethics. Even in like primates, you cannot do any research you want in monkeys and baboons and higher primates. In laboratory setup, the highest we can go is, earlier it was cats. People used to do a lot of neuroscience on cats and dogs. But then because cats and dogs are very, very, beautiful pets and people have affection towards them, the whole society stood up and said, okay, no more torturing cats and dogs. Go torture mouse and rats for the sake of science. So we now have descended down till mice and rats. And it is working well because mouse and rats almost have similar physiology as humans. They also show all those diseases, including diabetes, cancer, Alzheimer's, dementia, blood pressure, which is hypertension, tumors, everything, and even mental syndromes like mental disorders like depression-like states, anxiety, all these things are shown by a mouse as well. So it's working well. So we do our research till mouse. Insects, we are allowed to do a lot of things more freely. We can We can alter their genome, we can change their genome, we can put inside their genome, we can take out something from their genome, silence a gene, overexpress a gene. All these things can be done in insects, fish, and these organisms. Yep. Does that answer your question, Iram? But the labs that work, let me tell you, if it's not an insect lab, like my research is on Drosophila, which is an insect, so we do not have to go through a lot of ethical issues. But even the labs which are working on mouse and fish, zebrafish or some other fish, they have to get a ethical committee's yes, like a approval. So you have to tell in advance your projects, and the committee will then decide whether you are allowed to use these organisms. Is your research actually serious? Will it give some output? So your output should always be beneficial to humankind. If you're just doing for fun, no one will allow you to use a mouse. And even if you violate the rule, you take a mouse, to keep it in your house and do some experiment on it, and someone gets to know, there can be legal actions against the person, or if it's a lab, against the lab and the scientist. So ethical issues do come into picture. Yeah. And I think they should be in place. It's a very good thing. Okay, so so far so good. So we finished the chapter with a lot of extensive talk. I'll just take 2 more minutes before we finish, and I'll give you a homework for Wednesday. And basically what I want to do, what I want you to do is, you have to find some references for structure of DNA as a molecule. If you understand this, half of chapter 6 is in your grip already. Okay, Mariam has something. Wanted to say that I'm really so sorry, because actually I said as, okay, cool. It's fine, it's fine. Okay, it's not a question actually, it's something Mariam wanted to say to me. Again, Rabia is saying something. We have not started chapter yet. Okay, cool. So Rabia is saying that the chapter 2, which is plant reproduction, we did not start because I wanted to finish human reproduction and genetics first. So how about we first do chapter 6, because it is directly connected to chapter 5, whatever we have studied till now. Now we are going into the details, molecular details of it. And before we come to evolution, we will finish chapter 2. Will that work for you, Rabia and others? Because, you know, different schools have different things, so we have to juggle a little bit. Will that work for you, Rabia? Are you there, Rabia? Okay, I think she's writing in the chat. Okay, cool. Thank you so much for allowing this. So what I want you to go and figure out is structure of DNA as a molecule, and also structure of RNA as a molecule. Okay, these 2 structures, just the structure. Look at the structure, what it is made up of, and some related information about DNA and RNA. You don't have to go into details, all that I will cover. And in the next class, the broader question of the next class is why we need a genetic material. Okay. So all this time we studied about genes, genetic inheritance, disorders, blah, blah, blah. But why do we have a genetic material? Do all organisms have genetic material, or are there organisms that are blessed that they don't have genetic material and they don't have to worry about all of this kind of inheritance thing? So try to find the answers to some of these questions, and I will see you all in the next class. Okay, we'll start with some biochemistry. So those of you who do not like structures or organic chemistry, I'm so sorry. I'll try to make it as easier as I can, but please do some homework before you come. Okay.
Yes, sir. Thank you, sir.
Okay, sir.
Okay.
Thank you, sir. Bye.
Bye bye. Take care.